Hi and welcome to this talk on Far-UVC for virus inactivation. My name's Ewan Eadie and along with my collaborators I've been doing a little bit of research and a big bit of reading into so-called Far-UVC radiation. Over the next 25 minutes I'm going to share with you what I have learned in these last 6 months. First, some disclosures, so I have no conflicts of interest to declare for this presentation. The presentation is going to be structured like this. I'm going to start with a little bit on sources of UVC, a little bit on efficacy, and then a big bit on safety. So let's get started. For this audience today, I obviously don't need to go into any detail on the electromagnetic spectrum, as I'm sure you're all very familiar with it. Our bread and butter in medical physics and optical radiation is the UVB and UVA portions of the spectrum. It's far less common for us to be involved with UVC. In fact, up until March of this year, I had very little involvement with or knowledge of UVC. I had calibrated a couple of UV detectors for customers against UVC lamps, and I'd seen the excellent presentations and posters from members of this community at past IPEM meetings. But my hospital doesn't use UVC, and therefore I really hadn't got involved in it directly. That all changed in March when the brother of a colleague sent me an email with a paper from Dr. Brenner's research group from Columbia University. That email was like a brainworm that I just couldn't shake, and which has ultimately led me to exposing my skin to greater than 500 times the exposure limit value. There'll be more on that later. But for now, I think it is worth just explaining a little bit about the UVC portion of the electromagnetic spectrum and the common UVC sources. So between 100 and 200 nanometers, UV doesn't travel very far as it's rapidly absorbed in the air. So we're not interested in that section of the spectrum. It's the wavelengths between 200 nanometers and 280 nanometers that are of particular interest because of their germicidal properties. Although there is still some germicidal action right up to 320 nanometers. Now in the UVC region, the main artificial source is the low pressure mercury lamp, which is very similar to the lamps inside our phototherapy cabinets. Our phototherapy lamps are mercury lamps, but they have a phosphor coating on the bulb. The mercury emissions hit the phosphor and produce the narrowband UVB or broadband UVA radiation that we're accustomed to. But if the phosphor is removed, then what remains are the very sharp mercury lines, mercury emissions, with the primary emission at 253.7 nanometers, as you can see on the slide here. These mercury lamps are cheap and they've been around for a very long time. It is well proven that they are effective at destroying viruses and bacteria, even drug-resistant bacteria such as MRSA. They're used in large-scale industrial applications, and of course these are the lamps that are in the UVC units that are used for cleaning hospital rooms, and which you may have already in your hospital. But as you also may know, these lamps must be used in unoccupied spaces because they will cause a painful erythema on the skin and a painful keratitis in the eye if you get overexposed, which really can happen very quickly as the systems are designed to be powerful in order to get effective decontamination. Now, as well as the low pressure mercury lamps, there are other lamps which emit UVC, which I haven't gone into here, both in the interest of time and because they're not the focus of this presentation. A technology which is available now, and the one which I am focusing on today, is the krypton chloride excimer lamp, which is the main lamp currently operating in this part of the electromagnetic spectrum, dubbed the Far-UVC or Far-UV. This isn't an official term, but it is being used to try and separate this wavelength range from the conventional germicidal mercury lamp. The krypton chloride excimer lamp has a peak wavelength of about 222 nanometers, as shown on the slide here. Now, without a filter, there's also some low-level, longer wavelength emissions, which are important, as we're going to see later on. There's a lot of hype about this particular lamp and about Far-UVC radiation, because the research thus far kind of indicates that the technology can inactivate viruses but not cause harm to the human eyes or skin. Now, if you just kind of let that sink in for a second, a technology which can decontaminate the air and surfaces in occupied spaces, literally killing the virus as it's breathed, coughed, or sneezed out. Of course, the true story is more nuanced than that, which is what we're going to go on to look at next. First, how does UVC actually inactivate viruses? Well, the primary mechanism is through the damage to the DNA or the RNA of a virus, depending on if it's a DNA virus or an RNA virus. And the UVC breaks the hydrogen bonds that hold a base pair together and cause cross-linking. Now, because the DNA and RNA are responsible for the virus replication, damage to these critical parts results in inactivation of the virus. The slide shows an example of one particular type of cross-linking. And in the past few months, there have been studies demonstrating that UVC is effective against the SARS-CoV-2 virus, which of course is the virus that's responsible for the current COVID-19 pandemic. In this study by Healing Low, they irradiated the SARS-CoV-2 virus, which had been isolated from a nasal swab of a patient suffering from COVID-19. They irradiated with both UVA and 254 nanometer UVC. And while both had some impact, the UVC was more effective. A dose of just over 1,000 millijoules per centimeter squared resulted in a 6 log reduction in the viral load. That 6 log reduction is generally regarded as being enough to state that the virus has been inactivated. Similarly, a study by Kitagawa showed a 2.51 log reduction at just 3 millijoules per centimeter squared. 2.51 log reduction represents a 99.7% reduction in the viral load, and that was actually the limit of detectability in the assay that they used, which is why they couldn't get any higher log reduction. So these are just 2 studies. There are a few others, and they show that in a laboratory environment on a surface that both 254 nanometers and 222 nanometer UVC can inactivate the SARS-CoV-2 virus. But what about inactivation of the virus in air? Well, UVC hasn't been tested specifically with aerosolized SARS-CoV-2. I'd imagine it's probably quite a difficult thing to do, particularly as you need a category 3 lab, but it has been shown to successfully inactivate other aerosolized coronaviruses, HCOV-229E and HCOV-OC43, as well as the H1N1 influenza virus. Now, given that viruses are generally more susceptible to UV in air than they are on surfaces, we can be pretty confident that the 222 nanometer UVC is effective at inactivation of aerosolized SARS-CoV-2. So with the evidence, it's clear that 222 nanometer UVC is effective at inactivating SARS-CoV-2, probably in air and on surfaces, which is not entirely unexpected. So the question that then remains to be answered is it really safe for use in occupied spaces? That is an incredibly difficult question to answer definitively. So let's take a look at the evidence, and at the end you can come to your own conclusions. The first study to look at the safety of 222 UVC in humans was actually from the photobiology unit in Dundee, where I'm based, and it was as far back as it was published in 2015. The study was a first-in-person, within-subject dose escalation study, which is basically means a series of MED tests, starting at low doses and building up, looking to see what dose the erythema appears. Now, based on some previous published lab data from other research groups, the investigators really didn't expect to see any erythema. The study was designed to be a small initial safety study, which was going to be a precursor to a much larger safety and efficacy study. However, of the 4 subjects that had been tested, the investigators found that erythema between just 40 and 50 millijoules per centimetre squared. The investigators found that there was erythema at doses between 40 and 50 millijoules per centimetre squared. Now, to put that in context, the ICNIRP exposure limit value at 222 nanometers is 23 millijoules per centimetre squared. So the MEDs were at doses twice the exposure limit, but they were also an order of magnitude too low for a bactericidal effect, which was thought to be around a few hundred millijoules per centimetre squared. In addition, when the investigators biopsied the volunteers, they found that DNA damage was not just in the suprabasal, but also in the basal layer. Why is that important? Well, in the uppermost part of the skin, the cells can't replicate. And so damage to those cells is not thought to be an issue in terms of skin cancer. But in the basal layer, DNA damage might lead to mutation and then replication, and then it is a skin cancer concern. So the results of this study really stopped any further investigations taking place, and they were a bit puzzling because up until that point they were kind of contrary to what had gone before, and as we're going to see, they were contrary to what was to come as well. Now, what followed were a series of studies all showing that the safety of 222 nanometer UV light, not in humans, but in the laboratory. So first, the study of Buonanno et al., which didn't induce CPDs or 6,4 photoproducts, which are just another marker of UV DNA damage. And they weren't induced in either a 3D skin model or in a group of mice. The mice received about 157 millijoules per centimetre squared of the 222 nanometers in a 7-hour period, which is a slightly higher dose than the biopsies in the Woods study. And then that was followed by a study by Narita et al., published in 2018. And in mice, they were irradiated with 450 millijoules per centimetre squared each day for a total of 8 out of 10 days. There were no CPDs and no swelling, no erythema was detected in the 222 nanometer irradiated mice. All of those effects were induced in the 254 nanometer irradiated mice, which was down as the positive control. So why then was there DNA damage and erythema seen in the Woods study from 2015, but not in the subsequent laboratory studies, which delivered much higher doses? Now, maybe the most obvious difference between the studies was that the last 2 slides I've shown were with mice and the Woods study was in humans. But round about the same time as the previous study I mentioned, there was another study which had been undertaken in 2018, but actually wasn't published until 2020. Fouquet et al. Investigated 20 healthy volunteers and exposed their backs to between 50 and 500 millijoules per centimetre squared of 222 nanometer UVC. And 24 hours after irradiation, they saw absolutely no erythema. The sites that were exposed to the 500 millijoules per centimetre squared were biopsied, and they did show some CPDs, which were slightly but significantly higher than in the non-irradiated skin. Now, there were a couple of things that weren't optimal about this study. First of all, from the original Woods study, the erythema that was observed arose around 5 to 7 hours after irradiation and had actually disappeared by 24 hours. But unfortunately, that fact didn't get reported in the paper. Fouquet et al. Looked for erythema at 24 hours, so they may have missed some of the changes in the skin. The CPD analysis also wasn't assessed for the different layers of the skin, and we know that we've already heard how important it is to know where the CPDs occur. But despite those limitations, the effects observed in this study from Japan were of lower magnitude than the Woods study and at much higher doses as well. So the mice versus human argument isn't the whole explanation. So why then the effects seen in the wood study? A study which was performed in a department that I work in, and which I know would have been very thoroughly conducted. So that was really the brainworm that I mentioned earlier on that had kind of taken hold of my attention and really just wouldn't leave me alone. Now, I'm very fortunate to work with some incredibly talented computer simulation experts at the University of St Andrews, Dr Kenny Wood and Isla Bernard, and I was able to pose the question to them to see if they could help address the problem that I was having. Now, with Monte Carlo radiative transfer modelling, they were able to take the spectrum of the lamp that was used in the Wood study in 2015 and simulate how the lamp's spectrum travels through the skin. And if you look at the graph on the left-hand side, what you can see is that the longer wavelengths that penetrate further into the skin, you can see that it's the longer wavelengths that penetrate further into the skin. If you remember earlier, I said we would come back to those low-level, longer wavelengths in the krypton chlorine lamp spectrum. And when you add in the CPD action spectrum to the computer model, it becomes obvious that it was definitely the wavelengths above about 240 nanometers that had been responsible for the majority of the CPDs in the Wood study. In fact, if you go back to the Wood study, there is a sentence in the discussion, and it says, there is the possibility that longer wavelength UVC contributed to the effect. So was the lamp spectrum used in the study by Woods different to all those other studies? Yes, it was. All the other studies used a filtered Far-UVC source, which intentionally removed those longer wavelengths, as you can see on the slide here. So it seemed that longer wavelengths were responsible for the erythema and the basal layer DNA damage that was observed back in the 2015 study. What we'd like to do now, of course, is another study looking in human volunteers at the effect of a filtered Far-UVC. Whilst we try to set that up though, I've been fortunate enough to be able to do some self-exposures to a filtered Far-UVC source. Now, based on the literature and the computer modelling, I was comfortable increasing my exposure to the filtered Far-UVC source. So I undertook multiple exposures on my inner forearm to varying doses of filtered Far-UVC, 4,000, 8,000 and 12,000 millijoules per centimetre squared. The source that I used for the exposure was a filtered Far-UVC source, as already mentioned, from Ushio, and the spectrum of which was displayed a couple of slides ago. The source had the characteristic peak at 222 nanometers and had an irradiance of 4 milliwatts per centimetre squared, which was determined over the wavelength range from 200 to 400 nanometers and was measured at the exposure distance. All measurements were performed on a calibrated spectroradiometer in an ISO 17025 accredited lab with traceability back to national standards. I observed the irradiated areas visually, which is a subjective measurement, but I also measured the area objectively with a reflectance spectrophotometer. And in the next few slides, I'm going to show you the results from these exposures. Now, it's extremely important, of course, to remember that this is an n=1 investigation, and whilst I made every effort to control all of the variables, it is definitely not a proper controlled trial, and it is subject to both bias and error. Although having said all that, let's see what happened. With all of the exposed areas, there was no erythema at any time point, either visually or by measurement. Now the graph here shows the change in A, where A is the green to red ratio from the spectrophotometer. A positive value means an increase in red relative to green. Now, as you can see, each of the doses was mixed in with the control site, and so this measurement technique couldn't distinguish any erythema, and I couldn't observe any erythema visually either. What was observed though was a yellow colouring of the skin, which was present immediately after exposure at all irradiation doses. Now the graph you see on this slide is the change in B, where B is the blue to yellow ratio, and a positive value indicates an increase in yellow relative to blue. So it's clear that all of the doses, delta B is initially increased compared to the control. Now, according to the spectral measurement, the pigmentation persisted in the 12,000 millijoule per centimetre squared irradiated site beyond 24 hours, but actually I couldn't see that visually. I wasn't able to see that pigmentation the next day. It was difficult to capture this yellow pigmentation in a photograph, and it may not display very well on the screen, but this image is of 2 irradiation sites, and hopefully you can see within each square the yellow colouring of the skin. So clearly some form of chemical change is taking place. The UV being emitted by the lamp is being absorbed by something in the skin, and some change is taking place which is causing the pigmentation. Now, without biopsies or laboratory investigations, we can only speculate about what is going on. And my speculation is that it might be proteins in the stratum corneum and the uppermost parts of the skin absorbing that radiation and causing the change in colour. There are a couple of more hints that support this part of the speculation. Now, the majority of the colour disappeared when the skin was tape stripped. Tape stripping is literally as it sounds. It's taking sellotape, placing it on the skin, and then removing the tape. And that was done 30 times. And as you can see on the graph, delta B drops right down to 0 following tape stripping. As tape stripping removes the stratum corneum, this would hint that the majority of the colour is in the stratum corneum, but not all of the colouring. There appears to be a bit of a recovery a few hours after the tape stripping, indicating that there is likely to be some penetration into at least the mid-epidermis, which is also what the computer modelling shows. It's also not entirely clear which wavelengths are causing the colouring. We initially thought it might be the lower emission UVA wavelengths that are present in the filtered Far-UVC source. But actually, when we put in additional filters to reduce the longer wavelengths even further, we still observed the colouring of the skin. So that indicates that it was most likely the 222 nanometer peak, which is being absorbed and causing that colour change. But remember, of course, that the doses that are being delivered and causing that colour change are several hundred times higher than the current exposure limits. Now, I'm based in a dermatology department, so I have very much focused on the effects in skin, but what about the eyes? Because the eyes are generally more sensitive, but there's much less research available. The one published study that I found demonstrated actually that there was no corneal damage in some rats at doses up to 600 millijoules per centimetre squared. Now, I'm also aware that there is a centre in Florida using Far-UVC to treat corneal ulcers, so deliberately shining on the eye. Although as far as I can tell, whilst they've presented their data at conferences, I haven't actually found a published study from them. Anecdotally, I've also heard that tearing in the eye occurs at doses much lower than 600 millijoules per centimetre squared, suggesting that there's maybe something akin to the skin happening also in the eye, where the energy is being absorbed very superficially and causing some kind of chemical change. Okay, we've come right to the end now, so let's recap on what we've covered. In this overview, we've seen that Far-UVC at 222 nanometers appears to be effective at inactivating the SARS-CoV-2 virus responsible for the current COVID-19 pandemic. It's not surprising and is on the back of decades of research of the germicidal properties of UVC radiation. The real question is, can it be used safely on a population? We already actually have a legal exposure limit, so it's okay to use as long as the dose is kept below this value. The animal studies which have looked at mice at mice who are susceptible to skin cancer are showing no increased risk in those mice. The human study did show a slightly increased CPD at doses far exceeding the current exposure limit, and there were no more CPDs than you would expect from about 20 minutes outdoors. And yes, there were visible changes to the skin at the very large doses that I used, and the visible changes seemed to be limited to the upper parts of the skin, where the cells can't divide, and so it's thought not to be a skin cancer risk. But I guess after having heard that all over the past 25 minutes, I'd like to ask you, what do you think? Can Far-UVC be used in occupied spaces? I'd love to have that debate with you in the Q&A, but I'll just give you my personal opinion before we go. I don't actually think Far-UVC presents a skin cancer risk. I think all the studies and the computer modelling show that 222 nanometer doesn't penetrate far enough into the skin to reach the critical cells in the basal layer. But the energy from the Far-UVC source has to be absorbed somewhere in the skin. Maybe in the proteins in the top layer, maybe in the bacteria that lives in the skin. And then what does that energy do once it's been absorbed? So we don't know. And we don't know what we don't know yet. So for sure, more research is definitely needed. But does the benefit outweigh the risk in this particular pandemic? I think probably in certain high-risk areas, such as maybe healthcare and care homes, it probably does. However, if it is only for air disinfection that we want this technology, then why not use upper room UVC? This technology has been around for 90 years. It's reasonably cheap. It's been proven effective. It's recommended by the World Health Organization for controlling tuberculosis transmission. And when it's installed correctly, it's perfectly safe, as the individuals that are in the room are never actually exposed. So why bring in a new technology when there is a fantastic existing technology without any safety issues? That question is going to have to wait till the next time, because I am out of time. Thank you for listening today, and thank you to all of my colleagues, collaborators and social media contacts who have been supplying constant information on the exciting new technology that is Far-UVC.